In our last discussion, we were talking about interferon and complement, and those are two that are considered to be innate. They don't require antibodies, but if we get to slide 70, the classical pathway actually does require antibodies, and the other two, the I can't say that word, alternative pathway and the lectin pathway are antibody independent. So they can just go on without needing antibodies nearby to start the whole process. All of the pathways end up at C3, and C3 makes C3A, which goes off and causes inflammation, and the C3B goes off and helps with immune clearance, phagocytosis, and poking holes in cells, cytolysis. So this is a whole lot of stuff. You don't have to memorize all of this stuff, but be aware that complements can work with antibodies, but they also can work separate from antibodies. Now, as I was looking through this and going into the inflammation thing, in order to have inflammation, you usually have histamine. So we know that mast cells, basophils, and neutrophils are involved with inflammation. And I just wanted to point something out that I thought was kind of funny because I try really hard not to do politics, and I do really hard not to put anything on my Facebook page that smacks of favoritism for one party or the other. But there was a uproar with the former president, and somebody had been telling him about that you can use Clorox to clean everything and it'll kill viruses. And he was saying, well, wouldn't it be nice if there was something, that would go inside the body, like Clorox or something like that. Like I said, I try to stay away from things like that. But had he known, neutrophils actually make the same chemical that's the active ingredient in Clorox. It's hypochlorite. So we already know how to do that in our body. So we actually do put Clorox in our lungs, but we do it with our neutrophils, so we have control over it. So apparently there were some people who thought it would be a good idea to drink Clorox, and it absolutely is not. It's what neutrophils use to kill nearby cells, including themselves. So not a good idea, not recommended. So leave it to the professionals, leave it to the neutrophils to know how much Clorox to make and how much to release. And I was just kind of laughing, and I said, wouldn't it be interesting if neutrophils were able to sue Clorox for stealing their chemical formula? So, but neutrophils don't have a patent on it. And while we're talking about things that can kill you, remember Reye syndrome? Don't give children aspirin. If they have the flu or chickenpox or some of those viral things, we don't know exactly which ones. We do know that flu and chickenpox can trigger Reye syndrome in children. So if you're less than 15 years old, take Tylenol. Do not take aspirin. So I just want to point that out again. None of us enjoys feeling pain, but remember the prostaglandins and the bradykinins that cause us to feel pain, there's a reason for it. Pain makes you stop using your body part and give it a chance to heal. So that reminds me a joke about the woman who went into the doctor's office, and she was in great pain, and so the doctor told her to point out all the things that hurt. And so she would reach up and she pointed at her cheek, and then she screamed out in pain. And then she reached over and she touched her arm and screamed out in pain, and reached down and touched her leg and screamed out in pain. And she said, see, I hurt all over. And the doctor said, yeah, you have a broken finger. So now you guys know why I teach college instead of going on the stage as a comedian. So now we're going to start the last part of the chapter, and here are our goals. This, by the end of my talk tonight, you should know all of these things. You should know about adaptive, which a lot of people call acquired immunity, and what part of it is cellular and what part of it is humoral. And I'll just go ahead and give you a little hint that the humoral part would be the antibodies, and the cellular would be the cells that make the antibodies. And how you can get active immunity and passive immunity, natural and artificial. We're going to talk about what antigens have about them that makes them antigenic. And we're going to learn 5 different kinds of antibodies. We're going to learn how the B and T cells mature. And then we're going to find out about antigen-presenting cells and a little bit about interleukins. So that's our program for tonight. All right, here are the 3 ways that the adaptive immunity or acquired immunity is different than innate immunity that you're just born with. It is systemic. So it is everywhere in your body, not just at your skin or in your nose or in your lungs. It's everywhere. It is specific. So it is one pathogen. And not only that, but usually it's only a part of one pathogen, not even the whole pathogen itself. And the best part of it is you remember. So you have seen it before, your body remembers, and you can quickly stomp it out if it dares enter your body again. So there should be no noticeable illness. That's one of the things they're worried about right now, is if you've already had it, or if you've had the shot, the vaccine for it, and you are exposed to it again, will you reach a high enough titer to be able to spread it before your body knocks it out? So that's one of the concerns where they're saying, well, if I've already had the shot, and I've already had the disease, why in the world would I continue to wear a mask? Because I can't get it again, and I can't give it again. The purpose of cellular or cell-mediated immunity is to actually go kill a cell. Kill a worm, kill a protozoan, or if it's a cancer cell, go kill it. And this is the cool thing. If something is inside of a cell, then the antibodies can't get inside the cell to attach to them to stimulate the complement pathway and in the other ways that antibodies use to get rid of pathogens. So cellular, cell-mediated immunity can actually just go kill the whole cell. So you don't need to get inside and get at the thing. But of course, if you start killing too many cells, then you're in trouble too. So it's better to limit it to a small area, get rid of it, heal it, and be done with it. So the cell can kill cells that harbor pathogens. They talk about humoral immunity or antibody-mediated immunity. They used to think that the fluid running through your body contained humors. And sometimes they talk about, well, somebody is in a bad humor or somebody is sick because they have bad humors in them. And so they would actually bleed the person. They would cut them and let the blood run out of them. And supposedly the bad humors would come out along with the blood. Well, of course, what you're doing there is you're weakening the person and you're making it harder for their immune system to work. So that didn't actually work very well, but it was very prevalent for quite some time. Antibodies are good for binding to things that are extracellular. So a virus, goes into a cell, makes copies of itself, and then it has to be released from the cell, either by budding off or by just blowing the cell up. So some viruses are very gentle to the cell and they just kind of pop out. It's a good strategy because you don't kill your host, and so you just keep making more viruses and making more viruses. But the ones that just blow the cell up are hopefully there's some cell nearby that they can go and attach to and go into. But So these things have to be outside the cell in order for the antibody to find them. Antibodies don't go inside your cells. Yeast, protozoans, and then you have some things like toxic substances, venoms, and allergens. So one of the interesting things about toxic substances or toxins, a long time ago, I was working on a project, and my job was to find out what kind of a test could we make against the toxin of E. coli. Because the problem is, if someone was really sick and you're looking to find out what causes them to be sick, and you run a test for E. coli, everyone tests positive because everybody's full of E. coli. You have it in your GI tract. Ladies even have it in our vaginas. It's very common in our body. And actually, most of the E. coli is very helpful to us. But there are some bad actors, and they give off a toxin. And that toxin is one of the deadliest things in the world, because not only does it kill little kids, but it also kills livestock. So my job was to find a test to figure out if somebody had the bad E. coli, the one that made toxins. And so I did develop a test for that. And if you're just dying to know about it, you can just type in E. coli toxin and Karen W. Drake, and you can pull the paper up on Google. So here's where I went and just typed in Karen W. Drake, E. coli toxin, and it pulled up one that talks about how the toxin causes things to happen in your body. And then here's the test for the radioimmunoassay for it. So I was really big into using isotopes because they're very, very sensitive, and you can detect things very quickly. So what we had to do was raise antibodies against that particular toxin, and we raised them in goats. In order to get active immunity, you actually have to be exposed to the pathogen. So if you get the disease, we call that natural active immunity. You naturally got the disease and you naturally grew antibodies to it. You can have artificial active immunity, and that's when you get a vaccination. So you produce your own antibodies and your own memory cells to remember that disease, even though you've never had the disease. And I think it's interesting because this book was written a couple of years ago, and it says a vaccine consists of dead or weakened, attenuated pathogens that stimulate the immune response without causing the disease. So that was true until the time of COVID. And we have a new kind of vaccine where we are injecting the messenger RNA that knows how to make the protein part of the virus, the COVID virus, the coronavirus. So that's kind of neat and new. And we rushed it through very, very quickly. But I have a feeling we're going to see a lot more of these messenger RNA because there's no dead virus. So you don't have to worry about, well, did the person in the laboratory really kill it? Or is there a few live ones in there? You know, I'd be thinking that if they were going to give me like an HIV vaccine or something. It's like, man, if there was even one or two of those viruses still alive, that would be scary. Or if it's weakened, then you get just a mild form of sickness. So, but this messenger RNA business, apparently most people are just saying it hurts. The arm hurts like you got a shot, which, of course, anytime you stick a needle in your arm, it's going to hurt like that. But they're not having a whole lot of repercussions. They're not having a lot of side effects for it. And then there's some that are so scary, the diseases are usually fatal, like tetanus, that we go get booster shots. And that just makes a few more memory cells just in case we encounter tetanus. So we want to keep our immunity high. So those are the two active forms. You either get a vaccine or you get the disease itself. But there are passive forms of immunity, and this is kind of neat. And you can go get antibodies from somebody who's already had the disease. So you just take their blood, you take the antibodies out, return their blood to them, and use the antibodies to inject into somebody who is sick but hasn't had a chance to make any antibodies of their own. Now, the good thing about this is it goes in and does everything right then. I mean, from that second on, it's in there, it's attacking, and it's wiping out the disease. And if you're just now getting sick, then there's not a whole lot of the disease to have to wipe out. So that's really cool. But you're not getting an active disease, and you're not getting the memory cells. So it is possible later on down the road to then go ahead and get the disease because you were exposed to it, but you wiped it out before your body actually had a chance to mount the whole immune response, because that takes up to, like 6 days to get that activity going. The other way that you have natural passive immunity is getting antibodies from the mother through the placenta, or once the baby is born, from her milk. Now, this is one of the things that really upset me when I was pregnant with my children, because someone somewhere had put it into the medical curriculum for nurses and for doctors that mother's milk was no good. And they said, you can tell by looking at it. Look at cow's milk. Cow's milk is all creamy and full of all that fat and calories for the baby to thrive on. And look at the mother's milk. It's almost blue, and it's really thin. So it's obviously no good whatsoever. And so nurses were actually trained before the mother, if the mother said, bring my baby to me, I want to nurse my baby. They would give the baby a bottle of water or a bottle of formula, and then they would bring the baby to the mother, and the baby would refuse to eat because the baby was full, and it'd just fall asleep. And I didn't realize that that was what was going on with my first child. But by the second child, they tried to do it again with my second child, and I said, oh, no. You bring the baby in here, and I'm going to nurse that baby from the get-go. And they just tried to explain to me that mother's milk just was not any good. And I was dumbfounded. So anyway, obviously they hadn't had a course like this where you learn that the mother's milk has all the antibodies to protect the child against every disease the mother has ever had. So the child, as long as it's nursing, will not get measles, will not get mumps, won't get any of the kinds of flu that the mother's had, all the way down the line. So mother's milk is the healthiest thing that you can give a baby, because it's not just getting fat and calories in the milk, you also need the antibodies. So hopefully they're not still teaching that in nursing school. So every time I teach any class to anybody that might be a nurse, I'm trying to pop that bubble. It's like, oh my goodness, don't believe it if somebody tries to tell you that, because it is so full of so many wonderful things to keep your baby from getting sick. And one more thing about human milk for human babies is we're programmed to make something that's easily digestible for our babies. So it's much more easily digested and less likely to cause gas and colic than would be any other form of milk or formula. All right, so that's natural passive immunity. As soon as the mother stops nursing the baby, then the baby is now susceptible to every single disease. So that's why while the mother is nursing the baby, it's a good idea to go ahead and give the kid the vaccinations for diphtheria, pertussis, tetanus, so the DPT, and now they've added some more like chickenpox and measles. So that's great. We're getting more and more vaccines for the children so that they don't have to go through the disease itself. Now, artificial passive immunity, this slide's a little bit confusing because right here it says natural passive is temporary immunity from antibodies from another person. Artificial passive immunity is temporary immunity from antibodies from another person. You're like, wait a minute. One of them's natural, one of them's artificial. What's the difference? Well, it doesn't have to be from another person. You can get antibodies from an animal. So antibodies are cross species, which is kind of cool. Not only that, but we can just train microorganisms to help us. Or we can just get some cells and put them in some food and let the cells divide and make the antibodies for us in a test tube. So that's why it's kind of artificial. But the natural one was where you just went to the guy or the girl that was going to be donating the antibodies because they already got over the disease, and they run the blood out, pick out the antibodies, and then put the rest of the blood back in. So all they're interested in is just picking out the antibodies. And we use artificial passive immunity for snake bite, that may be venom, botulism poisoning, rabies, tetanus, and so on. So if a person, for some reason, had never had a tetanus shot, but then they were exposed to tetanus, you don't have time for them to mount an immune response. You don't have time to give them a tetanus shot and wait for them to develop antibodies and a memory cell. So in that case, you would have to give them antibodies right away. Not everything is going to trigger your body to make antibodies. So here's kind of a little list of things. In order to be antigenic, in order to be considered an antigen, you have to be over 10,000 AMUs. So if you add up the weight of all the atoms, that weight of each of the atoms has to be over 10,000. That's what that means. And you need complex molecules. So you're not just going to have some simple fat or something like that that's going to trigger an antigenic response, which is good because that would be terrible every time you ate a Snickers bar and all that fat and sugar got into your bloodstream and you're all of a sudden like, oh my gosh, we're being invaded by fats and sugars. So it has to be really large. It has to be very complex. And some of the things that'll do it, this is kind of an interesting thing. Proteins, of course, we kind of figured that one out because right now we're making antibodies for COVID against the proteins that are on the outside of the coronavirus. But polysaccharides, so sugars, complex sugars, not just like table sugar, that you put in your iced tea or something like that. Glyco means sugar, so sugar that's stuck onto proteins, so glycoproteins, if they're large enough, can stimulate antibodies. And glycolipids, so sugar attaching to lipids, to fats, can make them antigenic. So one of the things that's really important is when you're making antibodies, your body, your cells have to know which of the large complex molecules are yours and which of the large complex molecules are foreign. So luckily our bodies are really good at that. But in a minute we're going to talk about some times where the body gets confused and instead of attacking foreign invaders, it actually attacks you. And we call those autoimmune diseases. All viruses present antigens. All bacteria present antigens. All worms, all of the protozoans, they are going to have certain regions that will stimulate an immune response. So we call those antigenic determinants or epitopes. So we know all of those things are going to be considered foreign, and we're going to be able to attack those. Sometimes we need to make antibodies against something, a foreign invader, but it's too small. It doesn't reach that 10,000 atomic mass unit weight limit. And so we call these things haptens. They're too small to be antigenic. So that toxic substance that I was telling you that the E. coli made that I had to make a test for, it had a double whammy. First of all, you had to pick out the good E. coli from the bad E. coli. But it also was only 18 amino acids long. So it was a very small toxic substance, but it was enough that if you got that particular disease, you could have diarrhea so severely and lose so much volume so quickly that it would kill you. So it kills, still to this day, millions and millions of very young children. So that was one of the reasons I was happy to work on that project and come up with a quick test, because you don't want you need to get an antibiotic specific for the bad E. coli into the child before they lose all of their fluids and die. So what we had to do is we had to take that 18 amino acid toxin that we had purified out, combine it with something that made it look as if it was larger. So you just attach something onto it so that now it's large enough to attract the attention of the antigen presenting cell. So it'll go and eat it and then stick a piece of it up through the cell membrane. So we were able to grow antibodies, or have the goats make antibodies against the toxic substance for us. And then we would just draw some blood from the goat and take the antibodies out. And it would detect anybody who had a stool sample that, give a little bit of stool diarrhea, and you just put this in there, and if it bound to it, then you know the person had been infected with the E. coli that made that toxin. So, as I said, it was a shitty job, but someone had to do it. Some other haptens that get in your body, and some people have an allergic reaction to them, and other people are just fine with it. Some of the cosmetics, some detergents. I personally can't use Coast soap and Irish Spring. I think Irish Spring smells wonderful, but if I try and put that on my skin, I break out like crazy. So my body is able to take some part of that soap, even though it's a hapten, and make it large enough so that I get an allergic reaction. Poison ivy, some people are not allergic to poison ivy. Most people are. I personally am very allergic to animal dander. I can tell. I can walk into somebody's house and go, "Woo, you got mice." I know you do, because my nose just clouded up and I can barely breathe. And then penicillin. Most people have absolutely no trouble with penicillin. If you've ever seen a moldy orange, that kind of gray moldy stuff that's growing on the outside of a moldy orange, that's penicillin. So most of us don't have any problem with it, but some people, it can cause such an allergic reaction that it could actually kill the person. So, but it's small enough that it has to be able to bond to something else in order to get that whole allergic reaction thing going. I think I've mentioned this before, but it bears mentioning one more time to make sure you guys don't get confused. We've talked about hemoglobin. So globins are one class of proteins, and we're now talking about globulins. So globin and globulins, there's an extra syllable in there. And the different globulins that we have been talking about, the ones we call beta globulins, that's what complement is. Complements are beta globulins. When we talk about antibodies, we're almost always talking about IgG, or immunoglobulin G. Of course, because they're science people that are naming this stuff, they use the Greek letter, so it would be called gamma globulin, or IgG for short. That's what most people call it, or just antibodies. But you do need to remember there are other antibodies out there. We looked at this picture of the antibody before when we were talking about the complement binding region. And now we're going to look at it we're going to look at the constant regions. So in one person, when they're making antibodies, they always make this part and this part and this part and this part, the constant regions, the same way. The variable region is the one, and it's going to be able to attach to one part of a pathogen, one epitope, as they call it in this book. Immunoglobulin A, or IgA for short, is found, they call that the secretory because it's found in places where you secrete. So you secrete saliva, you secrete milk, you secrete tears, and you actually have intestinal secretions also. So mucus, saliva, tears are going to have IgA, this form of antibody in it. Now, what's really interesting about this, they suspect that this can protect you from getting AIDS. So one of the ways you don't get AIDS is kissing someone or drinking after someone who has AIDS. So they think it's a function of IgA. So that's a good thing. If you can figure out what protects you from something, then it gives you clues as to how to extend that so that it protects other parts of the body. The IgD, immunoglobulin D, or delta, if you want to use the Greek letter for it, is a monomer, meaning that it appears just as a single antibody, like this. Whereas we're going to find some of them will actually occur like with 5 of these. So we're going to talk about that in just a second. And this is on B cells. It is a transmembrane antigen receptor. So we think that IgD is important to activate B cells when they encounter an antigen. IgE, or immunoglobulin epsilon, is a monomer again, and it is a transmembrane protein. So it actually sticks through the membrane of basophils and mast cells. So if you trigger this, it's going to cause these cells, the basophils and the mast cells, to release histamine. And then you have the whole inflammation thing going on, the allergic symptoms, things like that. Plus, it also attracts eosinophils, which is kind of handy, eosinophils, if there's a parasitic infection. And it produces immediate hypersensitivity reactions. So a few people, just a very few people, have an immediate hypersensitive reaction to the COVID vaccine. And so anytime they give you a COVID shot, they want you to stick around for like 10-15 minutes and see if you have an allergic reaction. And they've got EpiPens, they've got epinephrine, which we call adrenaline, on hand in case you do have an allergic reaction. So immediate hypersensitivity reaction. But luckily, very few people have this. But and as far as I know, they haven't figured out why. It's just a handful of people who have that reaction. So a summary of what IgG, which is what we usually are talking about when we're talking about antibodies, are immunoglobulin G, or gamma globulin. It goes by many names, unfortunately, but IgG is what most people do it for short, or just call it antibodies. It's about 80% of all the circulating antibodies. So if I were to draw blood from you and I looked at your blood, I could see if you had antibodies against COVID. So that's what they do when they're talking about doing an antibody test to see if you've already had COVID. But the problem with that is the test that they are using can't always tell the difference by whether you have had this coronavirus or if you've had another coronavirus in the past, because coronaviruses have been around for a long time, causing just what we call the common cold. So it's hard to tell whether you're looking at antibodies from the common cold or if you're looking at antibodies from COVID, this new thing that's coming around. But that's why they draw a little bit of blood if they're looking to see if you've already been exposed. And then, of course, after you've had the vaccine, then in the experimental people, when they were checking to see if it actually was working, they would draw a little bit of blood and see if they developed antibodies against COVID. So IgG is the main antibody. It's about 80% of all the antibodies that are running around in your body. IgM is a pentamer. So this one would be a wheel of 5 antibodies. So it can bind to a lot more stuff a lot faster. And it is secreted in your primary immune response. So when you very first are exposed to something, you're going to make IgM first. And then as your body figures out what it's doing and you have this whole antigen presenting thing going on, and the B cells and the T cells are talking to each other, then you get your secondary immune response and you get your IgG. So IgM is first on the scene, and let me show you a picture. So here you go. These are the constant regions right in through here. So here's one. There's the 2 legs of it. Here's one, there's the 2 legs, there's one. So there's 5 of these in a ring, and each one of these is able to grab a hold of some foreign invader. So you can see how quickly it could mop up invaders. So you're going to see the IgM first on the scene. It's a pentamer, it's 5 of them. It's going to be in the plasma, and it's going to be in your lymph. So you remember we talked about the lymph nodes, and we talked about the white blood cells that are in the lymph node, and how the lymph goes in by afferent, but there's only 1 or 2, at most 3 exits out of the lymph node. So it stays in there for a while, and it gives the white blood cells a chance to get anything that's a foreign invader and then start the whole process. So you're going to see IgM being made in the lymphatic system really quickly, because that's where the lymph nodes are, and they're constantly screening for foreign invaders. For those of you who want to go on and study genetics, here's one of the things that they have different values in different books. So some of the books will tell you have 46 chromosomes, and each chromosome has about 10,000 genes per chromosome. And then you get a book like this, and it says there's only about 20,000 genes in all of our chromosomes that make everything that we are. And so, of course, one of the questions that people had was, well, then how can we code for all the diseases that are out there, all the bacteria, all of the viruses, all of the pathogens that are out there? Because the human immune system can make as many as 1 trillion different antibodies, but you only have 20,000 genes. So again, you have to look at which book you're looking at as to how many genes we have, but we definitely don't have 1 trillion genes. So they do somatic recombination. That means your body recombines just the end of the antibody. So DNA segments can shuffle just a little bit, making new combinations to produce antibody genes. So you don't have to reinvent the wheel. You don't have to have a whole gene in order to make a specific end on the specific antibody. So this has been really a curious thing, and they've been really looking to see how we're doing this. So we don't make new genes. We just do this shuffling of DNA segments. So all the cells you got to remember are the B and T lymphocytes, and then the antigen presenting cells, which they call APCs. And macrophages are really good at it, dendritic cells, and there's some other antigen presenting cells that we'll run into as we go along. You're going to find these cells in the lymphatic system, especially the lymph nodes, and any organs that you have in there, the spleen. You're going to find the dendritic cells out in your skin. So that's your first line of defense. Something gets in past through the dead layers, the dead outer layers of your skin, and get down to where the cells are alive and they're full of food and nutrients that the foreign invader would like to get a hold of. And so it's nice to have dendritic or branched cells that have their little fingers sticking out everywhere in your skin to encounter any foreign invader and then sound the alarm. And then you're going to have them in mucous membranes. So we talked about your lungs, your trachea, the whole respiratory system is full of mucous membranes, and your intestines from your mouth all the way through your anus, the digestive system full of mucous membranes. Everybody agrees that the B and T lymphocytes are born in your red bone marrow, and in you, because you're an adult, it'll be in your flat bones, not in your arms and legs anymore, because that's turned to fat. And we think that the T lymphocytes leave and go to the thymus where they grow up and learn how to do T cell things. And the B lymphocytes are released already mature and knowing how to be a B cell. So all they have to do from that point on is decide whether they're going to become a plasma cell or a memory cell. And I'll explain that process in just a second. Now the T cell, it's got to go and decide, well, is it going to be a helper T cell or a regulatory T cell? So it has a few choices to make. But remember, old people don't have a thymus, but they do have immunocompetent T cells. So there's a little mystery going on there. Just keep that in mind. So, this is whoever was typing this up. It's supposed to be born in bone marrow, but I guess they just got real excited with all the B's and just kept on. So the T cell is born in the bone marrow and educated somewhere, probably the thymus, at least when you're young. But the big thing that I want you to know about that is you need the T cell to develop surface antigen receptors. So it's got to have a receptor sticking up so that when one of those cells, an antigen presenting cell, for example, a macrophage, I told you the macrophage will grab something, tear it into pieces, and stick those pieces up on its cell surface. And when this T lymphocyte comes along and detects this foreign piece sticking up out of the antigen presenting cell, like a macrophage, then it knows to glom onto it and start the immune process. How do you like that scientific word, glom onto it? I probably should have said hydrogen bond to it and be just a little bit more scientific. Your book spends some time talking about the maturation process of a T lymphocyte, but one of the things that I'm going to say quickly is the T cell, once it matures and it has its little receptor there that's able to find antigens that are presented to it, it needs to be checked out by the thymus or wherever it's maturing and make sure it doesn't react to you, to self-antigens. So if you don't, then this T cell can mount an immune response to your own tissue. And we definitely don't want that to happen. So you have a mechanism in the thymus that will take those cells that can't tell the difference between self-antigens and foreign antigens, and it'll kill them. It's kind of interesting that only about 2% of the T cells actually only find foreign antigens. The other 98% can't tell the difference between you and a foreign antigen, and it has to be destroyed. So what this slide says is the B cells are ditto. Some remain in the bone marrow. Some of them go through clonal deletion. If they can't tell the difference between self and foreign, they have to be killed, the same as the T cells. They put surface antigen receptors on so that they're mature and ready to go. And then they go out the bone marrow and colonize the same lymphatic tissues and organs as the T cells. So the B cells and T cells have to work together for this whole immune response to do its thing. The T cells can do some things without B cells, but not very much. So the antigen-presenting cells, the APCs, are the dendritic cells in the skin, the macrophages, which used to be monocytes but grew up to be big and angry, and we called them macrophages at that point. Reticular cells, which are usually found in the lymph nodes, and some of the B cells can present antigens to the T cells. So the function of the antigen-presenting cells is depends on major histocompatibility complex proteins. Now, these are fascinating, and if you want to read more about them, feel free to do so. These are some of the proteins that make you. So you have major ones, and you have some that aren't so important. And histo, that's tissue. So these are major tissue compatibility proteins. So if you are going to give me a kidney, I'm going to give you a liver, whatever, we need to match up with as many of these major histocompatibility complexes as we can. And I can't remember if I talked to you about the fact that these MHCs actually give you a smell. So people can actually respond to you because of the major histocompatibility complexes you have. So some people, just everybody likes them. You just notice that. It's weird. They don't have to know them. They don't have to know if they're good people. They're just attracted to them. And other people, it's not that they're not clean. It's not that they smell like a locker room or something like that. You just get close to the person, you just don't really like them for some reason. And you don't really have a reason not to. And so they've traced it back to major histocompatibility proteins. So I'm always thinking, if I ever decide to have another career, I had to start a matchmaking a store, company, online, something, where you send me your cell sample and I'll look at your major histocompatibility complexes and match you up with somebody who has compatible MHCs to your MHCs, so that you would fall instantly in love because of your compatibility. These major histocompatibility complexes are used to label every cell of your body as belonging to you. So that's what you're going to make sure that the B cells and T cells understand that and don't go and attack you. But another thing you can use these for is to find baby daddies. So if you say, well, that's the guy who got me pregnant, then you can look at his major histocompatibility complexes and the baby's major histocompatibility complexes and get paternity to where it's, undeniable. Whereas if you just look for blood type, there's only 4 blood types, so, you can't rule out somebody as the baby daddy just using blood typing. So other than making you and making you smell attractive to some people, the MHC is a place where the antigen-presenting cell can put the antigen, the epitope. So it sticks it up through the cell membrane and is held in place with the major histocompatibility complex. So I went out on the internet to see if I could find a picture, and this purports to be a macrophage, and you can see it pulling in bacteria. So it's pulling them in, it's sending out these part of its cell membrane to grab a hold and pull it back in. And then it's going to digest it, and it's going to put little pieces of these bacteria back out on its cell membrane. And then a T cell will come along, and it will be attracted to these major histocompatibility complexes, and it will see that there's something there that's foreign. So there is a docking and an indoctrination of, hey, this is what you need to go out and fight. So this is the antigen presenting cell, and then there's the T lymphocyte. Now, this one they've labeled a macrophage, but it probably is not, because macrophages are gargantuan. And they're like 4 or 5 times the size of a lymphocyte. So I'm going to think that this is a smaller antigen presenting cell that they just said, oh, it's a macrophage, but it really isn't. Here's a better picture that I got off the internet. You can easily see that the T lymphocyte, which we're going to call a helper T cell, is much, much smaller than a macrophage. Significantly smaller. So a number of macrophages, a number of helper T cells can be serviced by one macrophage. So they all come in, they all see a little piece of whatever the foreign invader looks like, and then they go off. So, but this is a really busy slide. So, but I want you to see that there's a substantial size difference. This is kind of a busy cell or slide, talking about the cytotoxic T cells, also known as killer T cells, also known as CD8 cells. And then you have helper T cells, and these are going to be really important, helper T cells, in understanding AIDS. They promote, they talk the killer T cell into killing things, and they activate B cells. So they're very helpful. And regulatory, once you've got an immune reaction going, so you've got the antigen presenting cell that tears up the foreign invader, gives pieces of it to the helper T cell, shows it to the helper T cell, and activates it, so it knows now what to make antibodies against. And then the helper T cell is activating the cytotoxic T cells and the B cells, so that the B cells are the ones that are actually going to make the most of the antibodies. But now you've knocked out the disease, so you've gotten over it, and you've maybe been sick. Usually if you get the flu, it's about 2 weeks, give or take a couple of days. So during that time, you've got this whole process going on, but when it's time for it to be over, something has to stop the reaction. So you have regulatory T cells, and they basically go around and go, okay, stop. They inhibit the division, cell division. They stop the cytokine secretions. They limit the immune response. It's like, okay, the war is over. We won. Everybody go back and stop dividing. So the helper T cells, depending on who's naming them, can be called CD4, CD4 plus. So there's a lot of stuff out there. You guys don't have to worry about that. Just remember that there are cytotoxic T cells that blow up cells. There are helper T cells that go get the information from the antigen presenting cell and bring it over to the B cell and turn them on. And then once the war is won, you've got other T cells, the regulatory T cells, that go along and shut the whole immune system down. And then, best of all, you have memory T cells, and they remember that exact illness, that exact flu, worm, whatever it is. It remembers the epitope. It remembers a part of the invader. So if that invader ever comes back again, the memory cell doesn't have to go and do the whole process. It just immediately kicks the immune system into high gear from day one. You don't have to wait 6 days to get the whole process rolling. Here's a cute little mnemonic you might want to remember. The cellular immunity is, first of all, you got to recognize it as a foreign invader. Then you have to react. You're going to have the antigen-presenting cells. You're going to have the formation of antibodies. And then you're going to make the memory cells. Now, I have a question that I don't know. Like I said, I'm reading as much as I can on the internet, and it's hard to know what's scientific and what's just somebody's opinion about things. But it seems like you could take the T cells that understand they have already been exposed to COVID and grow them up. You know, like I said, we can grow cells. We just give them some food and put them in an incubator, and we can grow cells. So why don't we grow a lot of T lymphocytes that know how to deal with COVID and inject those into people? But I've not heard of anything. The only thing they do is they look for the memory T cells that remember that the person has already had COVID. So I have seen a little bit about T cells that way. So a macrophage or an antigen presenting cell can find a foreign invader anywhere. You may have stepped on something and got something in your foot. You may have inhaled something. You may have eaten something, gotten in your GI tract. But you've got antigen presenting cells everywhere. As soon as they've found a foreign invader and they've processed it, they go to the nearest lymph node and drop off the information to the T cells. And the T cells feel the major histocompatibility complex that has the piece of antigen, or they call it an epitope, sticking out, and then it is able to start the immune response. I'm not going to test you over this, but as you're reading along, if you get confused because you keep jumping from major histocompatibility complexes to a different one, this is what's going on. Your major histocompatibility, which we call the type 2 proteins, are on the only on antigen presenting cells. Sorry, my tongue's getting tangled up. Okay, so you only find those, they label them 2, and they only display foreign antigens. So the cytotoxic T cells only respond to the major histocompatibility complex 1 proteins, and the helper T cells only respond to the ones on the antigen presenting cells, the APCs. So here's a slide showing you the antigen presenting cell. In this case, it's probably a macrophage. And here's the helper T cell, usually, although it could be the cytotoxic. It comes in, finds the epitope sticking out, finds the antigen sticking out, and then there's a co-stimulation protein. So you have a double connection here. And once you have this double connection, or co-stimulation as they call it, now the cell is activated and immediately starts doing mitosis and making copies of itself. So it knows what the invader looks like. It knows what the antigen is that it needs to carry the message out to the world. So here we go through the clonal selection, and you have to have mitosis. Now, mitosis is a slow process in eukaryotes because we have a nucleus. So it can take up to 24 hours for mitosis. So that's one of the reasons why it takes a while to get the immune response going when you're first exposed to something that you've never been exposed to before. Now, some of these helper T cells, some of these cytotoxic T cells will continue on and go and talk to the B cells, or they'll just go out and find a cell that's infected and blow it up. But some of them are going to go off and be memory cells. So they're going to stay out of the fight. They're not going to join in. They're not going to go out and stimulate other cells. They're just going to remain in your bloodstream, hopefully for the rest of your life, remembering what this antigen looked like. So we'll spend a second talking about cytotoxic T cells before we move on to the other B and T cells, the helper T cells. So they dock on to a diseased cell. They're the only type of T cell that actually hits a whole cell. And this would be like a virus-infected cell or one that's been overcome with bacteria. Once it attaches to the cell, it's going to poke holes in it. It's going to put enzymes inside the cell, and it's going to kill cells the same way as the natural killer cells do. It's also going to help with making interferons. And remember, interferons are made by the dying cell, and they go out to nearby cells and cause them to mount a protection so that if the virus tries to come inside the cell, it's already ready and it stops it before it can start reproducing. And you also have tumor necrosis factor. This is seen in cancer cells. So not all tumors are cancer. There are benign tumors, but the cancerous tumors, the ones that can metastasize and spread, we want to kill those. So it's good that the tumor necrosis factor will aid the macrophage activation. So it will alert the macrophage that there are cancer cells in the area, and then it'll help kill the cancer cells. And some of the white blood cells, like the neutrophils, they give up their life fighting the good fight. They'll kill some bacteria or the invader, and then they'll release the Clorox chemical and die because it kills everything in the area, including them. But the cytotoxic T cell, it just pokes a hole, puts some chemicals in there, sends some other chemicals out there, and goes off looking for another enemy cell, leaving the dying cell behind. Here's a picture taken with an electron microscope. All pictures made with an electron microscope are black and white. So they just colorize them so you can see the different things of interest. So these are T cells, and they are actually poking holes in this cancer cell. So it's interesting to see how big a cancer cell is, or body cell is, compared to the little T cells. So these are cytotoxic T cells, and they're literally poking holes through the cell membrane. And you can tell that this cell's dying because it's blebbing, B-L-E-B, blebbing. It has these little things where the cell membrane is kind of bubbling up, and that lets you know this thing's about to pop. So we've spent some time talking about the 4 different types of T cells: the helper T cells, the cytotoxic one, the regulatory one, and the extremely important memory cells. So now we're going to talk about how the helper T cell activates the B lymphocytes. After the helper T cell has activated the B lymphocyte, the immunocompetent B cell now puts surface receptors for the antigen that it was exposed to. So now it's only looking for just that one epitope, that one part of a virus or that one part of a bacteria. And when the B cell finds that particular antigen and it sticks onto the receptors, this will get the B cell activated. All those antigens will be linked together, so you have a big mouthful, and you pull all of those antigens inside the cell. So we call it receptor-mediated endocytosis. This is a cartoon drawing that somebody made, but this is an actual picture taken through a transmitting or transmission electron microscope. So here you have the cell membrane right here, and you're going to put proteins, receptors, out here. If you find the antigen, it's going to stick in here, and it's going to be pulled inside the cell. So this is endocytosis, or cell eating. And here you have a little sac with some of these receptors and some of the antigens. So here's an actual picture. Here's the cell membrane. You can see the cell membrane. And you can see that it is pulling in all of these receptors, and you can see the antigen that it's pulling in. So it's kind of neat. It doesn't tell us what it's pulling in, but this is endocytosis. After the B cell has pulled some of the antigen in, it's going to digest them, it's going to pull them apart, and it's going to put some of the antigens on its major histocompatibility complex 2 proteins. And at this point, it'll just stop. But if a helper T cell comes along, it can bind to it. It can secrete chemicals called interleukins, and that will cause the B cell to start dividing. So it knows what the antigen looks like because it ate some. It actually took it in by endocytosis, and the helper T cell activates the B cell. The B cell starts dividing. Most of them are going to be what we call plasma cells, and the plasma cells are absolutely crazily cranking out antibodies to the point where the cell will wear itself out and die. It will work so hard, it won't stop to take care of itself, to eat what it needs to eat, and do the things it needs to do. So plasma cells literally wear themselves out in 4 to 5 days. So some of the B cells that know what the antigen looks like and that were stimulated by the helper T cell are going to go off and be memory cells. So just like the T cell had a subset that goes off and it stays out of the war. It knows what the enemy looks like, but it's staying out of the war, and it's sitting over there with the information. And the B cells do the same thing. So we have memory B cells and we have memory T cells. So that if you ever encounter that antigen again, that virus, that bacteria, you immediately know what to do with it. And you don't have to go through the whole antigen process. You're ready to rock and roll. So here's a flow sheet telling you what's going on. And so here's your B cells, and they've got their little receptors out there. They pick up an antigen, they pull it inside, they process it. The helper T cell comes along and says, yep, that's a foreign invader, all right. Here's some interleukin, and the interleukin causes the B cell to start dividing, and then you get clones, identical cells, and some of them are going to continue on and become what we call plasma cells. And the reason in this picture that they've got this kind of pink stuff is that's endoplasmic reticulum. So if you're busy cranking out stuff really, really fast, like the B cells are cranking out antibodies, you need a lot of endoplasmic reticulum. You need the ribosomes that are sitting on it, so this is rough endoplasmic reticulum, and you need to be able to then secrete these antibodies. So they're making it and sending it out to go out and do the humoral immune response. So these plasma cells are making these antibodies that circulate throughout the whole body. And then a few of them become the memory cells. These are actual pictures of just a B cell. There it is, got its nucleus. And then here's one that's been activated. And look at the endoplasmic reticulum. And a lot of mitochondria. It's going to take a lot of energy to do this process. But they'll wear themselves out and die fairly soon. And you have 3 or 4 ways that the antibodies can then stop the infection. So one of them is literally just stick on to whatever it is that's in your body. Literally coat it. So this prevents it from being able to go into a cell. It prevents it from it immobilizes it, because you've got so many of them that are stuck together that now they can't go freely to go throughout the body and infect. So they get stuck in the lymph nodes usually. Another thing you can do is you do the complement fixation. So you've got the IgM, which is the one that's got the 5 subunits, and then the IgG, which comes on a little bit later in the immune process. Once an antigen binds to the active area, the ends of the IgM or the IgG, remember they can change shape, they can expose the place where complement attaches onto the side of them, and then you can get rid of things through the processes that complements do, such as inflammation, phagocytosis, immune clearance, or tearing open a cell. So complement fixation is like against foreign cells coming in, worms, things like that, bacteria. And if somebody accidentally gives you a blood transfusion that doesn't match, or if they try to transplant a kidney into you and you don't have it's not very compatible, then you're gonna destroy it. Here's a cartoon they drew trying to show you what would happen if you gave somebody a transfusion of the wrong type blood. So here you have those little 5-sided IgM, and you can see that this one is holding on to this red blood cell, this one's holding on to that red blood cell. So you've got these two linked, but this one's also attaching these two together. And so what you get is a big blood clot. You get a big clump. So that obviously is not going to be able to go through your capillaries, and they're going to be destroyed because as they try going through the capillaries, they'll be like a log pile. They'll just start popping. So agglutination and precipitation using antibodies. The more of the antigens that you can get a hold of, the bigger a blob you get, the less likely it is to be able to move, and the more easily a macrophage coming along or any of the other cells that can eat, that can do phagocytosis. This is a big mouthful, so they can get all of those antibodies and antigens in one mouthful. So a quick overview of what's going on. We have the primary immune response. This is the first time your body's ever seen whatever's invading it. And you're going to have to go through that whole process of the helper T cells, going over to the macrophages, the antigen-producing presenting cells. And this could take anywhere from 3 to 6 days before you finally are cranking out massive amounts of antibodies. First thing on the scene, IgM, and after about 10 days, you stop making the IgM. IgG takes a little bit longer to come onto the scene, but it can last much longer than the IgM. So you continue making IgG after the IgM is kind of pooped out and not making any more. If you're exposed to the same illness again, the plasma cells are formed within hours, not days. And the IgG goes quickly. So you don't mess around with the IgM. You have some, but mostly you're just kicking out the IgG and away it goes. And it says the response is so rapid that the antigen has little chance to exert a noticeable effect on the body. So that's a fancy way of saying if you've had it before, you'll not get sick with it again. And one of the things that people are always saying to me is like, well, then how come you get the common cold over and over and over again? Well, you don't. There's over 200 different organisms out there that cause the common cold. Anywhere from rhinovirus, adenoviruses, coronaviruses. So you don't get the same cold over again. But if you get one that's related, then you do get over it more quickly because the memory cells may recognize some antigens that are similar to something they've seen before. For those of you who like graphs, here is showing where you first primary response are exposed to something. Here's your IgM coming on. By day 5, you should start feeling a whole lot better because you've got a lot of IgM being made. Remember, it's got 10 spots that can grab hold of an antigen because it's a 5-plex or a pentamer. And then after about the 6th day, now you're cranking out the IgG and it continues on. So within a month, you should be completely well. And here's what happens if you see the same antigen again on day 0. The first day, bam, you got that IgG going. So you have a much higher titer than you had before. And then there's your IgM. And now we're going to spend some time talking about what happens when the immune system runs amok. So if things go correctly, it's a beautifully choreographed thing. And then after everything is well and you've conquered the invader, then the regulatory T cells come in and shut everything down and you go back to feeling normal again. But sometimes your immune system has what we call a hypersensitivity. You overreact to whatever the substance is. So here is, we're going to talk about anaphylaxis. And you can have localized anaphylaxis, which we don't really care that much about. But if you have anaphylactic shock, this is where you have released so much fluid from such a hypersensitivity reaction that you actually can't keep your heart pumping and you die. So anaphylactic shock is definitely a hypersensitivity reaction that we need to know about. Other hypersensitivity is if you're doing everything you think you're supposed to be doing and you have your self-tolerance fails. Now, a classic example of that one is strep throat. So kids are forever getting strep throat. And you know, if you've ever been tested for it, they take a cotton swab and they stick it down and they kind of run it over your tonsils, which usually gags you like crazy. So I always hated that. But then they can check and see if you have the streptococcus or if you have strep throat. If you do, it's really important to get an antibiotic and knock it out as fast as you can because there is some part of strep that apparently looks a little bit like the proteins in your heart. So as you're busy making antibodies against the various bits and pieces of the streptococcus, you may be making antibodies against or they will be recognized as the parts of your heart. And so the antibodies will go out and mount an immune response against your heart tissue. And we call this rheumatic fever. And it can leave the heart damaged or can lead to death if it destroys enough of the heart. So some infections, childhood infections like strep throat, are more dangerous than just the disease, but only in people who mount a hyper sensitivity reaction. So not everybody who gets a strep throat gets rheumatic fever, but it's not everybody who takes aspirin gets Reye syndrome and ends up with their brain swelling until it crushes it and they die. So it's good to know when weird things happen. 2 words you guys need to know are chronic and acute. Acute is something that just comes on quickly. One of the most common acute illnesses is when you eat something that doesn't agree with you, and then you spend a lot of time in the bathroom either vomiting or having diarrhea or doing both. So that is acute. It comes on quickly, and it usually resolves itself in a day or two. Whereas something that's chronic just goes on and on and on and on. So if you've got arthritis, that's a chronic condition. If you get bronchitis, where you're sick and then it seems like it settles down in your lungs and you just cough and cough and cough for months, those are called chronic. So if it's long lasted, it's chronic. And you're going to deal with it in a different way with your immune system than you do with the acute one. Now, we talked about the different types of antibodies. So we've got IgA, IgM, IgD, IgE, and the IgE is the one that is on the membrane of the basophils and the mast cells. And anytime you see the word basophil or mast cell, especially mast cell, think, oh, whoa, histamine. So you're going to have that whole fluid rushing into the area and histamine reaction. So IgE is on the membrane of the basophils and the mast cells. As soon as something that can bind to the IgE binds to it, then the cells will start secreting histamine and you'll get the whole inflammatory reaction, chemicals being released. You can get glandular secretions, vasodilation, your capillaries open up, your fluid rushes into the area. The smooth muscle in the area can start having spasms. So it says it usually subsides within 30 minutes, but if you have a really strong hypersensitivity, it can actually go on to be severe or fatal. So bee stings, peanut allergies, things like that. So what does it look like in your body? Well, you got swelling. You are going to hypersecrete mucus, so your nose starts running or your eyes start dripping or your skin starts swelling up with fluid underneath it. You we call them hives. And if it's internal, then like you ate something that didn't agree with you, you get stomach cramps, diarrhea, vomiting. So food allergies and asthma would be examples of this type 1 hypersensitivity. We call this anaphylaxis, and if it's in just one area, you can take antihistamines or you can buy like Benadryl cream to rub on the area. But remember, anaphylactic shock, this is where it gets into the bloodstream and you're having this allergic reaction throughout the body. So it is much more severe than what we were talking about before. So you have constriction of your airways, labored breathing, widespread vasodilation, circulatory shock, which can lead to death. So if you open up your blood vessels enough that the fluid runs out into your tissues, you don't have enough fluid to keep the heart pumping. So, but you can usually tell somebody the bronchoconstriction and the labored breathing, you know that they're having some severe problems. So these are the ones you give the shot of adrenaline or the EpiPen, epinephrine. So if you're in anaphylactic shock, antihistamines aren't going to take care of the whole thing. So you can give them antihistamines, but by the time the antihistamines kick in, the person's probably already dead. So epinephrine opens up the bronchioles, opens up your airways, increases your cardiac output, and gives you back your blood pressure. And of course, if you've lost a lot of fluid into your tissues, then giving the person something to drink will also help hydrate them and put fluid in their circulatory system to keep the heart going. This constriction of your airways is known as asthma, and it's the most common chronic illness in children. So chronic means that they have it, get over it, have it, get over it, but it's plagues them. It's not like something where you have 24 hours and then you're all better. Usually they're allergic to something. If you can figure out what they're allergic to, then it'll stop the asthma. It can be triggered by emotions. So sometimes if a kid gets really upset, they'll have an asthma attack. Too much exercise can also cause an asthma attack. So a kid who's having an asthma attack, or an adult, usually adults can take care of themselves, but little kids don't know what's going on, and so they don't know exactly what to do. But they start having spasms of their bronchial tubes. You can hear them coughing and wheezing, and if it swells up enough, they can actually suffocate. They just literally can't get air down their airways to get to the lungs. So asthma is one of the things that kills little, kills little children. You can have a rebound or another respiratory crisis a little bit later, because remember, the immune system kicks in and it knows that there's something wrong, but it takes it a little while to get this whole process going. So sometimes after you've gotten the patient stabilized 6 to 8 hours later, then they have a second crisis. So you've got eosinophils being attracted in the area. You've got the respiratory cilia. Now, when we do the next chapter, we're going to talk about respiratory cilia. So you guys, you don't know exactly what we're talking about, but your trachea is lined with these little cilia, and when a person is having an allergic reaction, they stop moving. They're paralyzed, and all the mucus and stuff that's being made because of this hypersensitive reaction is going to run down into the lungs. So that's not a good thing. The epithelium, the lining, can be scarred and turned into scar tissue, which could lead to long-term damage to the lungs. So asthma is not something that you poo-poo away and say, oh, no, the kid will be okay in a little while. You need to treat it very seriously and check and see, do they need epinephrine, or are they going to be able to start breathing on their own? Type 2 hypersensitivity can usually be avoided. It's because somebody gave you the wrong blood, or somebody gave you a drug that you have a reaction to. So I can't take morphine. If you give me morphine, I will vomit and vomit until I literally don't have enough energy to vomit anymore. So that really sucks, because when you have surgery, one of the first things they do is they give you morphine when you wake up to relieve the pain. And in my case, they can't do that. Type 3 hypersensitivity is more chronic, it's more long-lasting, and it's usually an autoimmune disease. So here's two of them. One of them is lupus, and if it's systemic, it's a little more scary. Sometimes it just affects the kidneys, and sometimes it's throughout the whole body. Lupus gets its name because they said that it looks like a person who's having an attack of this autoimmune disease is wearing a fox mask. So lupus is Latin for fox. When we do the renal system, when we talk about the kidneys and the formation of urine, we're going to learn that the active unit, or the part that actually is filtering the urine, is called a glomerulus. And we're going to find, whenever you see the word itis, you have an inflammation. So occasionally your body runs amok and attacks your own glomeruli. And so obviously that's not a good thing. So those are type 3, and it's antigen antibody complexes that precipitate out into your blood vessels or other tissues. So that's why that person had the red rash on their face. You get complement activation, you get inflammation, the person has a fever. And the last type of hypersensitivity we're going to talk about is type 4, or delayed sensitivity. So you put on some makeup, you've never worn that makeup before, and then the next day you wake up and your face is all broken out. You're walking out in the woods and you don't realize that that thing that has 3 leaves on it is poison ivy. And you wake up the next morning and you are scratching. Usually it's in the middle of the night. You wake up just scratching at yourself and you get these blisters all over your skin. So if you have a TB skin test, they'll inject something just slightly subcutaneously under they don't inject it into the muscle, they don't inject it into your blood vessels. They literally try to put it just underneath the skin and see if you have a reaction to it. And if you do have a reaction to it, within a day or two, you have to come back and have them look and see, did it just go away, or did it puff up, turn red, and show that you have antibodies already to TB. So if you have antibodies already and you mount an immune response within a day, we know that that's a secondary, and we know that you've already been exposed to it. So that's why they give you a TB test, usually before you start a new job. Definitely, like if you're a teacher, they want you to have a TB test before you're out there teaching kids and spreading tuberculosis to them. And unfortunately, there is a type of flu. If you are genetically programmed to have type 1 diabetes, you may be fine for years. You may be 4 years old, 10 years old, and then you catch this one strain of the flu, and while your body is trying to get rid of the flu, you mistakenly think that the beta cells in your pancreas are foreign invaders. And as you get over the flu and kill all those viruses, you also kill the beta cells that are in your pancreas. And now for the rest of your life, you'll never be able to make insulin again, and you're going to have insulin injections for the rest of your life. Unless, of course, we get this stem cell research going, and then we should have a cure for it, because you can grow new cells. If you want to watch a movie that takes your heart and stomps on it, watch Boy in a Bubble, starring John Travolta. There are some people who are born without B cells or T cells, so they can never mount an immune response using antibodies. So and they have very little innate immunity. So for the rest of their life, from the time they're born until they die, they have to live in a bubble, literally, that filters out any possible pathogen because they cannot protect themselves from it. It's almost like you're being born with AIDS, where AIDS, if you contract the HIV virus, it gets into your body, it goes into your bloodstream, and it finds helper T cells. So there's a receptor on the surface of helper T cells that invites the AIDS virus inside. And the AIDS virus divides inside the helper T cell, and over time, it will spread and wipe out all the helper T cells. And we hopefully have learned from this process that you need the helper T cells as an intermediate to make the antibodies. So the person who gets AIDS doesn't die from AIDS. AIDS is not a fatal disease by itself. Usually you're sick for a couple of weeks, feel like you just had a mild case of the flu, and you don't even think anything about it. But then you start getting other diseases, and you realize that what happened was now, if you've knocked out the helper T cells, you can't fight off other diseases. So you're going to die of pneumonia, you're going to die of cancer. So opportunistic things that you normally would not die of, you now can't mount an immune response. So some people are born without B and T cells, and they end up having to live in a bubble for the rest of their life. And others who get AIDS end up slowly wiping out their immune system as they go lower and lower and have fewer and fewer helper T cells. So here's an actual picture of a kid who had to spend his life living in a bubble. One of the things that I want to point out emphatically at this point in time, so you guys don't make the same mistake that a lot of people are making. AIDS is a scary disease. It is a retrovirus. It is an RNA virus. And the RNA is reverse transcribed. It is RNA, and it is reverse transcribed, so it makes DNA. And that DNA can actually combine with the chromosomes in your body. So it is scary. The coronavirus is also an RNA virus, but it is not a retrovirus. It does not replicate and make DNA. So it is a mild, usually, disease, unless your body is hypersensitive to it or you have an existing condition. You're already dying of lung cancer, or you're already dying of congestive heart failure, something like that. So a lot of people are thinking that this coronavirus is a retrovirus like AIDS is, and it is not. You can get over it. Whereas the AIDS virus has gotten into your helper T cells and is knocking out your immune system. So completely different infectious routes and prognosis. So if you hear somebody saying, oh my goodness, this coronavirus is just like the AIDS virus, say, oh no, it isn't. The AIDS virus is a retrovirus, and the coronavirus is just a normal RNA virus that's not reverse transcribed, turned into DNA, and attached to your chromosomes. Here's an artist representation of HIV, the AIDS virus, and it looks a little bit like the coronavirus. It is circular. It has protein spikes sticking out. It has RNA inside. But this is a different kind of RNA, and it also has this reverse transcriptase, which the coronavirus does not have. Now, I wanted to point out something. When AIDS came out so long ago in the 80s, we didn't know what it was going to do. We didn't know how it was spread. There was so much misinformation and fear and confusion about it. But one of the things that they made a legal ruling, and I understand why they did it, but it is, I think, a mistake. So this is my personal opinion I'm giving you right here. But when you first have the HIV, you get infected with HIV, and they say you're HIV positive, you have the virus in your body. It is replicating. You are contagious. So you can pass it on through bodily fluids, having sex or sharing needles or getting a blood transfusion. Not by kissing, not by hugging someone. It's not spread that way. So you actually have to mingle bodily fluids. Now, unfortunately, a lot of nurses would wash their hands so much when they were working with AIDS patients that they had literally raw bleeding hands. And I've seen a lot of that with nurses who are dealing with COVID patients. So they have raw bleeding hands from washing their hands so many times that the body just can't cope with it. And so nurses were getting AIDS from working with AIDS patients because their hands were so chapped. So of course now we know that it would be better to wear gloves than to keep washing your hands over and over again to prevent the spread of AIDS. So if your body starts killing the helper T cells, when they take a blood sample, they can see that you have lower number. When it reaches a certain level, then they say, okay, you have lost enough of your helper T cells, you've lost enough of your lymphocytes, that we now can say you have AIDS. And the reason they do that is because you could walk up to someone and say, do you have AIDS? And the person is HIV positive, and they can look you right in the eye and say, no, I do not have AIDS. When in fact they have the organism, they have the virus in their body, and they can spread it. So they did it because if people found out you had AIDS, they would fire you in a heartbeat. So you could, with a straight face, say, no, I don't have AIDS. And so you could actually continue working, having the AIDS virus in your body for years before your white blood cell level dropped down to the point where you legally have to say, yes, I have AIDS. And the only reason I'm upset about that is because people are experimenting with sex. And if you ask somebody, before we have sex, do you have AIDS? They can say to you, no. And they may genuinely not know that they're infectious. So that's one of the things I worry a little bit about that. So if you want to know if someone has AIDS, say, are you HIV positive or do you have AIDS? And I don't make the discrimination between HIV positive and AIDS in order to encourage people to get fired or anything like that. I just do it because you need to know, even though you're just HIV positive, that you are contagious. Wearing a condom will stop the transmission of AIDS. Having blood tested before you give somebody a transfusion in the hospital will keep you from giving blood from someone who has AIDS to someone who doesn't have it and causing them to have it, catch it in the hospital. Another thing that people with AIDS should know is a lambskin condom as opposed to a latex condom. The lambskin has large enough holes in it. The sperm can't get through, so you can't get pregnant wearing a lambskin, but you can still get AIDS because you get the fluids and the virus is small enough to go through the holes that are in the lambskin condom. Used to, if you got the HIV virus in your body, if you got AIDS, then you usually had about 10 years to live. Younger kids usually didn't last as long because their immune system wasn't as robust as an adult's was. But now we have things that block the retrovirus, they block the reverse transcriptase, and we're coming up with more medicines now. So there are people who have AIDS who can live a full lifespan, so they won't die early. So it's not an automatic death sentence like it used to be, but you have to be very good about taking your medicines, and they're very, very expensive. So not everyone can afford them. So your initial symptoms of AIDS are flu-like symptoms. You have chills, you have fever. At night, you may wake up and your bedclothes are sopping wet because you sweated so much. You're just exhausted. You may have a headache, and you may have extreme weight loss. You may have swelling. Your T cell count, this is the one that tells you whether you're HIV positive or whether you're AIDS. Normally, your helper T cell count is anywhere from 600 to 1,200 for every microliter of blood that they draw. If you drop below 200 cells per microliter, then you officially have AIDS. So before that, you can say that you're HIV positive. The people who get below 200, now they're going to get toxoplasmosis. That's something that you can get from changing a cat's litter box. All of us carry pneumonia in our lungs, certain things that will cause pneumonia, and we're fine with it. It just stays in there and we keep it under control. But if you don't have the helper T cells, if you can't mount an immune response, then you can get pneumonia and pass away from it. The herpes viruses, we think about genital herpes, we think about the cold sores, those are herpes, but also chickenpox is herpes, and shingles. So there are different herpes viruses, and you will no longer be able to keep them under control. If you get tuberculosis, you can't mount an immune response. So people who have AIDS sometimes die from tuberculosis. Most women know immediately about yeast infections, and you can get yeast infections in other places. You can get it in your mouth as well as your vagina. So people who have AIDS a lot of times will have white stuff growing on their tongue, and it's a yeast infection. And then there are other diseases that are virally transmitted, and one of them is Kaposi sarcoma. They used to think that the AIDS virus caused Kaposi sarcoma, but it doesn't. It's a whole nother virus, and it's just the people who have AIDS often have also contracted the Kaposi sarcoma virus. Normally, people don't have any problem with Kaposi sarcoma, but if you're immune compromised, then you start growing these purple tumors on your skin. So old people, sometimes their immune system is so poor and they've been exposed to the Kaposi sarcoma virus that they grow these tumors on their skin. They don't have AIDS, they just have the Kaposi sarcoma virus. But there were so many people who presented with both that they were confused for a while and thought that the AIDS virus caused this, but it does not. How to get AIDS? If the mother has AIDS, it can pass to the baby. It doesn't always do it, but it can. If the mother has AIDS, she can pass the virus through her milk to the baby. And then, of course, everyone knows sharing blood and semen and vaginal secretions. So the most common way of catching it is to have sex with somebody who has it. Another way that's not as common is to get contaminated blood products. Especially people who are experimenting with drugs, they'll share needles. And if any one of the people sharing the needles has AIDS, it's very easy to pass it on with the needles. So you do not transmit it by casual contact. If we could figure out how to keep the AIDS virus out of the helper T cells, if we could find something that would block the binding site, then we could stop AIDS that way. If we could stop reverse transcriptase, we don't need reverse transcriptase in our body. It's the invading viruses that have this enzyme that are able to invade us and cause seriously bad diseases. So if we could block that. So of all the medications that we have and all the different ways that they work to try and stop the destruction of the helper T cells, none of them yet can cure it. We don't have a cure for it yet. We don't have a vaccine for it yet either, because it keeps mutating. All of the medicines that we have are, they have a lot of side effects, but they can extend your life, like I said now, to where you don't die after 10 years, you can live a full life. So, there is a lot of information. Good luck plowing through it. I've tried to hit the high spots. You need to go back and look and see what is the complement, how does it work, how are antibodies made, what are memory cells. But I hope you find it interesting and you're living through one of the more interesting times, because we now have a worldwide pandemic of something that should be very mild and is killing old people at a really high rate.